So, but you may know the number of microbes present in your system. You may know the type of microbes present in your system, but you still can't tell whether they are active or they are dormant in the system. So you need to know. It is best if you can measure their activity, but usually this is very process specific, which means uh, you you should be targeting a certain process, whether nitrogen fixation, respiration, production, because it's quite impossible to measure, to know everything about the activity of a microbe at once at one time. Okay, there's just too much information to be processed. Okay, so usually when you're measuring microbial activity, it is uh, process specific. Okay, so here is stated here that microbial abundance alone does not reflect microbial activity because abundance is the product of both production and loss. So the main point here is, if you see microbial abundance increasing in a system, if you're measuring microbial abundance in the lake, for example, and you see it increasing over time in 1 week, okay, you can't really tell whether the increase is because of production or because of less loss. Okay, so you can't really tell whether the increase in abundance is because of an increase in production or a decrease in loss. The only way you can determine it is if you've already also measured the production and loss. From there, then you can extrapolate whether which process is more important to determine the microbial abundance. So microbial activity should be measured in order to understand the functioning of the microorganism, whether the microorganisms are actually active and what function or role they play in the habitat. Okay. So when there are three common approaches that are used to measuring microbial activity. One measures the changes in substrate, the other measures the changes in a terminal electron acceptor, and then also changes in cell mass. Okay. For changes in substrate, it's either, for example, if you were to take a sample of water from the lake, you add glucose into it, then you measure the loss the changes in glucose over time in the in your incubation sample. Okay, what does that tell you about? So if do you think the glucose will increase or decrease over time?
Decrease.
Glucose will decrease
Okay,
after.
and why do you think it decreases?
Because it is used up by the bacteria.
The bacteria? How sure are you it's being used up by the bacteria? Not sure, right? Can you be sure?
Not sure.
Okay. So how can you increase your confidence that it is the bacteria that is using the glucose? What can you do to your sample?
we can isolate the bacteria, then undergo run glucose test.
Yeah, instead of isolating it, can you filter it? Because once you isolate it, once you grow it, then the bacteria changes. So instead of isolating it and culturing it, can we just filter the large organisms off? We can do that, right? Using filtration, maybe use a 1.0 micron filter. We remove the larger organisms. So in our lake sample, after filtration, the filtrate we will have only most probably bacteria. Okay. Then after that, we add glucose substrate, and then we can measure the glucose loss. So will that be a good indicator of bacterial activity? Anyone? Liu?
Yes, doctor.
So do you think yes or no? Yes.
Yes, yes.
But is glucose common in the environment?
I have no idea.
So glucose is a bit too rich for the environment. Okay, it's too labile. It's easily utilized. So although it may show glucose usage by the bacteria, it is not reflective of what's actually happening in the environment. Okay, so you can use glucose, but then maybe you can say this in your measurement is glucose uptake by bacteria, but it doesn't reflect the actual microbial activity because uh, it's just too rich, it's too labile, it's too easily available. Okay, that's why in natural environments it's very difficult to find uh, uh, high concentrations of glucose. Okay, but it's easy to measure, and it's also used by all bacteria. So sometimes researchers use it to indicate uh, the activity of microbes. Okay. So any questions? Okay, one other concern when you're using glucose, you have to incubate it longer in order to see a significant decrease in the glucose concentration. Okay. But when you incubate things longer, then the bias from bottle incubations start coming in. So nowadays, in order to reduce error and bias from bottle incubation, they prefer shorter incubation time. But shorter incubation time, you can only achieve if you're using radioisotopes, okay, radio-labeled substrates. Okay, so now if you read the literature, you'll see that a lot of them will use tritiated thymidine or tritiated leucine as substrates for the bacteria incubation. So what they do is, for example, they'll just take a sample of water from the lake, they'll add uh, tritiated thymidine, okay? So after 1 hour, they'll filter the bacteria and then check how much of a radioisotope is incorporated into the bacterial biomass, which means how much radioisotope is captured on the filter itself. Okay, and from there they can estimate how active the bacteria is. So among the two substrates that is used, tritiated thymidine and tritiated leucine, Thymidine is usually more related to bacterial replication because bacteria requires thymidine for incorporation into the DNA, RNA. Okay, DNA. Okay, for incorporation into their DNA, so it's related to bacterial replication. Whereas tritiethylucine is usually incorporated into their enzymes, into their proteins, so it's more related to bacterial maintenance and survival. So if you measure concurrently both thymidine and leucine uptake, you'll see patterns of change related to whether the bacteria is actively replicating or the bacteria is just carrying out a survival mode, maintenance mode. Okay. So that's the one approach to measuring microbial activity by looking at how the substrate disappearance over time, okay, or substrate incorporation over time. So the other approach is by looking at the terminal electron acceptor. So usually we focus on oxygen because oxygen is easy to measure and we can achieve quite high levels of accuracy by measuring dissolved oxygen. So dissolved oxygen can be measured either using the membrane electrode method or the Winkler titration method. So oxygen is related, either you can measure it, okay. So oxygen is related to microbial process rates, especially respiration, okay. When respiration occurs, oxygen is consumed. So a decrease in oxygen is related to respiration. Okay, but oxygen change can also be used to reflect photosynthesis. If you remember, oxygenic photosynthesis, if oxygenic photosynthesis is carried out, oxygen is released. So there will be a oxygen increase, okay, in your, okay, can you see this? Okay, so
Yes, doctor.
Yes.
this is one way of measuring a simplified method if you want to know what's happening in a system. Okay, basically what you need is just one light bottle and one dark bottle. Okay, you can either paint the bottle black or you can cover it with aluminum foil to keep it dark. Okay, so one set of light and one set of dark bottles. Usually what we use dissolved oxygen bottles, DOD bottles, because these are airtight and you can measure the changes in dissolved oxygen quite well in these bottles. So they are incubated outside, I mean in your environment under the sun. So the theory is in the light bottle, both photosynthesis and respiration occurs, whereas in the dark bottle, only respiration occurs. So in the light bottle, because there's photosynthesis, the dissolved oxygen will increase over time. Whereas in the dark bottle, because of respiration, the dissolved oxygen will decrease over time. Okay. So the changes in oxygen, dissolved oxygen over time in the light bottle is actually reflective of your net primary production. We use the term net because the changes in dissolved oxygen here is what you observe after respiration. Okay, so respiration is also involved here. So what you're observing here, any increase in dissolved oxygen reflects the net primary production. Whereas in B, in this bottle here, what you see here is the dark bottle. What you see here is the respiration. Usually they call it community respiration. Okay, so this is the decrease in dissolved oxygen over time. So if you take this rate and you minus off this rate, A minus B, what you actually get is what gross primary production. Okay, so this is the gross primary production is the primary production before respiration. Okay, so from this experiment itself, you can obtain quite a lot of information of photosynthesis and community respiration. Okay, so it is quite useful. Main problem is it takes at least 12 hours, 6 to 12 hours to get significant increase and significant decrease in dissolved oxygen. And it is also not really applicable in temperate countries. When temperatures are lower, it's harder, and sometimes you have to incubate maybe 2-3 days. But once you need to incubate it more than 1 day, there is just too much bias from bottle incubation. That's why it's not really practiced in temperate countries. However, in tropical countries, we find out that you can actually get significant rates even within 1 day, half a day. Okay, so that's why we use it sometimes to obtain data on photosynthesis, primary production, and respiration. Okay, but there's still a lot of unknowns because there are still some because even if you measure this, you do not really know which of the organism is actually photosynthesizing. Okay, and sometimes because there are changes in rates when you keep things in the dark and keep things under light, and because this bottle is more active, other processes are also different. So you can't really say that the differences here is only because of photosynthesis and respiration, because grazing, virolysis, all these processes also change. Okay. However, uh, the main principle for light dark dissolved oxygen method is this. Okay, the differences between photosynthesis, respiration, and respiration only. Okay. Any questions? Okay, so we'll move on. Okay, so
No,
with
Doctor.
yeah, yeah. Ada soalan? Any questions? Noah? Okay.
No.
So we've already talked about the substrate disappearance, terminal electron acceptor. Okay. So other than the uptake of substrate, okay, other than the uptake of substrate when you incubate samples, if you're looking at growth, you also see that there should be an increase in cell mass, or there should be an increase in cell abundance over time. This indicates microbial growth. Okay. So as I mentioned earlier, If you take a sample of water from the lake and you look at the microbial growth over time, you have to remove the effects of mortality, remove the effects of grazing, because bacteria in the environment is actively grazed by protozoa, protists. Okay, so to remove the grazing pressure, usually we will use filtration, size fractionation, 1 micron or 2 micron filter to remove the predators, and then we incubate it, and then we can see the changes in bacteria over time. Okay, and that will indicate bacterial growth. Okay, so for bacteria we can see an increase in cell abundance, whereas for phototrophs we can see an increase in chlorophyll A. Okay. However, like for sometimes when we look at the microscopy, when we look through the microscope, we also realize sometimes the increase in cell abundance does not equate the increase in cell mass, because sometimes the cells grow in size but they do not replicate. So you have to take into account whether if you observe that your cells are growing in size but not really multiplying, then you have to measure the cell size. That will be a better reflection of the increase in cell biomass over time. Okay, and so instead of depending on measuring cell abundance alone, you have to look at the cell biovolume. That will be a better reflection of cell growth over time. Okay, even for phototrophs, if you use chlorophyll A as a proxy, you have to take note that some phytoplankton has a higher chlorophyll A content in its cell versus others. So you have to take into account, because sometimes chlorophyll A is just easy to use, but it doesn't tell you accurately which cells are growing well. Techniques, you'll know about fish, okay, fluorescence in situ hybridization. But fluorescence in situ hybridization looks at the DNA, okay, but you know that the presence of a gene doesn't mean it's being transcribed, doesn't mean it's active. To see whether it's active, usually you need to focus on the RNA. Okay, that's why there is a technique called ISRT fish, in situ reverse transcriptase fluorescence in situ hybridization. This one looks at the RNA to see whether the gene is actually being transcribed, whether the gene is actually active. Okay. So for this ISRT fish, there are several pre-treatment DNAs to remove the DNA. This is for the sample. RNAs to remove RNA for the negative control, and a positive control with no pre-treatment. And then you use reverse transcriptase to amplify the RNA to change it to cDNA. Okay, and from there you use oligoprimers to look for the specific gene that you're looking for. Your primers are of course labeled with fluorochromes, and then you can observe under epifluorescence microscopy, and the cells that fluoresce are those that are actively transcribing the gene. Okay, any questions about this? Any questions about ISRT fish? Any questions about ISRT fish? Okay. Okay. So,
No, doctor.
other than using ISRT fish to look at the whether the gene is active, there are also qPCR methods that you can use, quantitative PCR methods that you can use to determine the activity of the gene.
The the cDNA or RNA?
Or
cDNA.
cDNA. Because the reverse transcriptase has already transcribed the RNA to cDNA. So you're looking at the cDNA. Any other questions? Okay, if there are no questions, we'll look at the quantitative PCR, qPCR. I'm sure you're also familiar with this. Are you all familiar with qPCR? Have you all done it in instrumentation?
We have learned it.
Okay,
Yes.
so I'll just briefly go through this. So real-time PCR is a major advancement in PCR technology, because with real-time PCR we are able to quantify the original template of the gene that you're looking at. Okay, so real-time PCR works because we are able to detect real-time the changes that's happening, the number of amplicons that's being produced over time. Okay, and we can do this because of the advancement in the imaging technology. Okay, the CCD sensors, the image sensors are sensitive enough to be able to detect the small changes in the amplicons that are being produced. Okay, that's so when real-time PCR came up, they realized the plateau stage is not reliable to do an endpoint quantification because of the variability here, but the precision is more at the exponential stage. Okay, so for real-time PCR, you can either use the cybergreen to intercalate the DNA and to quantify the changes. Okay, this is the cheapest approach, or you can use the FRET approach. Okay, a TechMan probe. Okay, a FRET approach, but this is expensive because you need to work with the supplier to come up with the specific probes to use. Okay, So what you need to know is probably this one. So you know that the quantification is at the exponential point where it breaches the threshold cycle CT. Okay, it means how many cycles it takes to breach the threshold base, the level. So at that, so this is actually correlated to the amount of original template. So so the amount of target amplified is proportional to the starting template. Okay, so at this point here, the amount of the CT when it is amplified is proportional to the starting template. So if you have a standard curve where you dilute your DNA others, and then you can plot the standard curve. From there you can estimate your starting template. So this is the usual parameters that they use. The baseline is determined from the first 3 to 15 cycles, or at least 2 cycles before the exponential phase. So from here, the baseline, they find out the standard deviation, and then the threshold is chosen as 10 times the standard deviation above the average baseline. Okay, and from there, once you set the threshold, whenever your line crosses the threshold, that will be your that will tell you how much is the starting template. Okay, just from there, you can know how many copies of gene do you have, and if you're looking at RNA, you will know whether the gene is being actively transcribed. That's how you can quantify your gene activity. Not either your gene activity or your gene copy number. Then for calibration, usually it's about 7 orders lower from the original concentration. We reduce it to 7 order. That will give you a good standard